let's get to understand proteins. Maybe we need to know what are proteins really. So proteins are biomolecules, meaning they are living molecules that are made up of carbon, hydrogen, oxygen, nitrogen, and in some cases they can also contain sulfur. Just like when you're talking about carbohydrates, carbohydrates are made up of carbon, hydrogen, oxygen. Now for proteins you also get to add the nitrogen, in some cases also sulfur. All right, so these are the elements that are going to make up proteins. Well, proteins are very much diverse. There are a lot in all living things, and they actually make up about 50% of the dry cellular mass. So very, very important. And just like carbohydrates have got monomers, they are made up of monomers or monosaccharides. Proteins are also made up of monomers, but the monomers of proteins are called amino acids, meaning that when you've got a lot of amino acids coming together, they are going to give you proteins. So out of all the biomolecules, one of them that does not have monomers is a lipid. So lipids don't have monomers, and so they don't have polymers. But carbohydrates, proteins, enzymes are also protein, and so they also have monomers, right? Now, what are the functions of proteins if we need to understand? Let's look at the functions. So, since there are a lot of kinds of proteins, some of them are used one in structural provision. Now, what do we really mean by structural provision? Meaning that they are found in structures that get to protect the body. For example, keratin. Now, this protein, uh, let me not say keratin, but collagen. Okay, this protein collagen is uh, is found in even in muscles, so even in the skin. So it is a very, very important protein that is used for structural provision, like in the skin. So it is very, very important. Collagen is present there. Apart from collagen, okay, for structural protection, we also have some proteins which are used in transportation. So they are there for transport. We can talk of the common hemoglobin, which gets to transport blood. So this is a protein, hemoglobin. Apart from that, we also have some other proteins which are enzymatic, as we had said, right? So we said we also have enzymes as biomolecules, which are made up of proteins. An example that we can give is a protein that is going to break down sucrose is called sucrase. So this protein is enzymatic in nature. Apart from this, we also have some other hormones which are there in our bodies, and those are also proteins in nature. An example that we can give is insulin. If we have a lot of carbohydrates in our body, those have to be stored, so we need insulin to store them. All right, so we need insulin to get to convert, to break it down, so that we will not have that diabetes mellitus. It's going to be converted to the glycogen, and if need be, it will be broken down again from glycogen to the glucose again. Right, very, very important function. Well, apart from that, other proteins are used for protection. We can think of antibodies. So antibodies are proteins which are there to make sure that we are protected from sicknesses and a lot of things that get to come in our bodies from the foreign environment. Other proteins are used in signaling. We call them signal proteins, right? So these signal proteins get to carry messages around the body. For example, you are going to talk of just insulin, which is a hormone which is involved in glucose level control. So it is there for signaling, to give signals. You need glucose to be broken down. You need glucose to be stored, and this thing and this thing. All right. So that is in cell signaling. Other proteins are used in storage. Okay, we are also have storage proteins. For example, I'm going to talk of albumin. So albumin, the protein store, okay, that is found in the eggs. That is a protein. It is there for storage. All right. So the proteins have got a lot of functions. We've talked of protection or defense. So all these are the kinds of functions we can still add on and on to the list, and they continue to go on like that. So these are some of the functions of proteins. Now we are saying that proteins, well, are made up of amino acids. Since we know that proteins are made up of amino acids, then we need to understand an amino acid. What really is an amino acid? What is its composition? Now, an amino acid is made up of an alpha carbon. This is called an alpha carbon. Now, to this alpha carbon is attached four different things. So, on one side, you have got a carboxylic acid functional group. On the other side, you have got an amine functional group. On the other side, you also have an ara group, which we call alkyl group, and then you also have an hydrogen atom. So all these are attached to the alpha carbon. Well, this group is carboxylic acid. This is what gives proteins their acidic nature. This amine group is basic. That is what gives proteins their basic nature. Now, this ara group differs from from one amino acid to the other, and it is the ara group that is going to determine what amino acid you have. Actually, there are about 20 different amino acids, and all those are determined by the kind of ara group which is present. So, out of these 20 amino acids which are there, some of them are essential, others are non-essential. Now, if you are talking of essential amino acids, these amino acids that the body really needs and the body cannot make them. So they are essential. You only get them when you get to eat plant plants, because it's only the plants which are able to synthesize essential proteins. The body is not able to make them. Well, as we now, when you're talking of non-essential amino acids, these are the amino acids which our bodies are able to synthesize themselves. Okay, so just this amino acid that we're able to see here, depending with what is on the ara group, that will determine the name of the amino acid. For example, the simplest amino acid is going to take the ara group is going to be an hydrogen. So when we see an hydrogen where there is R, that is the simplest amino acid called glycine. Right. So that is glycine, the very simplest amino acid. Now at the same ara group, when we see a methyl CH3, that becomes alanine as an amino acid. At least these this is simple. We should be able to know them. Like they are very simple to get to know. Okay, alanine, glycine. These are very simple amino acids, but the simplest amino acid is a glycine. If we get to consider carbohydrates, we said the most common form of carbohydrates is the D form, but for proteins, the L form is the most common. In other words, proteins are optically active, meaning that they can turn plain polarized light to the right or to the left. That means we can have D proteins and L proteins. These which turn plain polarized light to the left, this one turns light to the right. So the most common form is the L form, but for carbohydrates, the most common form is the D form. Right now, these proteins they exist in nature as neutral. So when they are in solution, these are going to exist as a zwitterion. So zwitterion meaning that they are going to have both positive and negative charges. But how does that get to happen? So this carboxylic acid functional group gets to interact with the amino group, forming positive and negative charges. And because of that, it is also called amphoteric. So something that is amphoteric is just something that is both positively charged and negatively charged. All right. So what is going to happen is that this hydrogen, which is here. Remember, from the definition of an acid is a proton donor, and when you're talking of a proton, is simply an hydrogen ion, and a base is a proton acceptor. We are saying this is basic, this is acidic. So this acidic part is going to donate this proton to the basic part there. So what we are going to have is this is not going to be there. Whenever something has got less than the actual number of oxygens, I mean hydrogens, it becomes negatively charged, and that hydrogen comes to attach here. When something gains more than the actual number of hydrogens it's supposed to have, it becomes positively charged. So this is how proteins are going to exist in solution as a zwitterion, having both positive and negative charges, or amphoteric. All right. So because of this, when you get to add an acid or a base, amino acids are called buffers. So that's why we say proteins are buffers. A buffer is simply a substance that makes sure that there is a constant pH. So a buffer is a substance that prevents a rise in pH or drop in pH. It makes sure that the pH is going to be the same. So when you get to add an uh, acid to a protein, or let me say to this amino acid, what is going to happen? Well, react this protein with an acid. The characteristic of an acid is that it's going to have an hydrogen ion. You can talk of hydrochloric acid. So this is going to dissociate, forming hydrogen ion and chlorine ion. Now this hydrogen ion is going to come and interact with this negative part. Why? Because the hydrogen ion is what? Is positive. So because of that interaction, what we are going to be able to see is that this charge is no longer going to be there. You are going to have an hydrogen attached here. When something has a charge of -1, you attach a hydrogen, is going to have a charge of 0. Attach another hydrogen, the charge goes to +1. Attach another hydrogen, the charge goes to +2. If you are removing hydrogens, the charges are going to be dropping like that. But if you're adding hydrogen, the charges are going to be increasing. So if we added 2 hydrogens, the charge would be +1 here. Okay, so we have added an hydrogen. That means we've introduced what? We've introduced an acid in this solution. So this is how an amino acid is going to exist in acidic nature. So in acidic nature, an amino acid is going to be positively charged. This is the positive that gets to remain here. That is in acidic nature. Now what happens if it is in basic nature? So just the same thing that you're able to see here, we can also write it as NH3 with a positive charge here. And then I'm going to put a dash, I'm going to put carbon. We can see we have got what? We have got an hydrogen. This is the hydrogen which is here. Or you are going to see we put an hydrogen down here and then the other group on top there. And then this side we have got a carbon. This is the carbon having 2 oxygens. One oxygen here, the other oxygen here, and an hydrogen. Okay, like that. So whenever you see an amino acid in this form, just know that this amino acid is in acidic condition. Okay, acidic condition. Why? Because you've got this hydrogen which has reacted from the acid. Now what happens if this amino acid is in basic condition? So remember, it is in the zwitterion form. So if it is in the basic condition, remember, this is already a negative sign. So the characteristic of a base is that it's going to have an hydroxide. So what is going to happen is this hydroxide is going to react with this part which is positive, gaining 1 hydrogen. Because of that gain in hydrogen, you are going to have this hydrogen out. So the charge is also going to drop by 1 because you've lost 1 hydrogen. And that is going to form a water molecule because you have got an hydrogen reacting with an hydroxide. So in basic conditions, the amino acid is going to remain with a net negative charge. So you can also write it as NH2, and then you have got a carbon, hydrogen, the other group, and then you have got COO with a negative sign. So whenever you see this, just know that the protein or the amino acid is in basic condition. I could as well put the R on top here and then the hydrogen down here. That means I would not have written this. So that is really the same thing. So we also need to get to understand this is because it's very important. So this is why proteins act as buffers. Why? Because they can react both with acids and bases. So when a protein gets to react with an acid, what is the charge that is going to have? When a protein gets to react with a base, what is the charge that is going to have? So we need to get to know that. Now let me just write the general. So here I've got a protein that's got an amine. Now I'm not going to write it in the Zwitterian nature. I'll just write it same way it's supposed to be. So here you have an amino acid. What happens when you react this amino acid with an a, an acid? Okay, an acid, the characteristic is an hydrogen ion. So what is going to happen when you react an amino acid with an hydrogen ion or with an acid is that this acid is going to react with the basic part, is going to interact with the basic part of the what, of the amino acid. That is what we're going to see now. That nitrogen is going to have three hydrogens. Okay, it's going to have three hydrogens like that. And is going to have a net positive charge. So because it's an acid, it's going to react with the basic part. You don't expect it to react with this acidic part, an acid and an acid. So it's going to have a positive charge when you react with an acid. And when you react an amino acid with a base. So here again another amino acid. Now the characteristic of a base is an hydroxide. What is going to happen? This hydrogen which is here is going to interact with the hydroxide. So in a case where, let's say we've been given water here, water is reacted with an amino acid. Now water itself can act both as an acid and as a base. Same, an amino acid can act as an acid and a base. So what would determine what is lost would depend on the conditions. So the amino acid can act as a base, water acts as an acid, or water can act as a base, amino acid as an acid, depending on the condition. Now when we get to react an amino acid with a base, this hydrogen is going to interact with this. So what you are going to form is a water molecule, and the net charge what you are going to have is you have got an hydrogen, another hydrogen. These are interacting with a nitrogen, carbon, hydrogen, carbon, double bonded with oxygen. We are going to have oxygen with a negative charge, and then you are going to have a water molecule that is going to be formed. So with a net charge of negative. So in other words, in basic conditions, you expect that the amino acid is going to have a negative charge. While in acidic conditions, you expect that the amino acid is going to have a positive charge. So this is also very, very important for us to get to know. It is very, very important. So again, I mentioned it. When you have got an acidic environment, the acid, what is going to happen? When you add an acid, the acid is going to interact with the what? The amine group. Because of that interaction, you are going to have a positively charged molecule. When you have got a base, okay, an alkaline environment or a basic environment, the result is going to be a negatively charged molecule. But in neutral conditions, proteins are not charged at all. They are not charged. So this is very, very important for us to understand, right? Now, just these proteins we are able to see, just these amino acids we are able to see, I've said one of their properties is that amino acids act as buffers, meaning that they neutralize either the acidic or the basic environment. That is the meaning of them acting as buffers. Well, at the same time, amino acids are going to react with each other to form proteins. So let me just put down three amino acids. And this is just a general because you can see the error group. I have not put anything there. That means I'm just using the general form. Okay, this is going to be the first amino acid. I can put down the second one. All right, and let me also now get to put down the fourth one, the third one, hydrogen. Let me just draw it much closer. Hydrogen here, hydrogen here. Okay, we've got a nitrogen, carbon with R group, hydrogen, carbon double bonded with oxygen, and then OH. Now what is going to happen is that these amino acids are going to be interacting. So this OH and this hydrogen, they are going to react, and this reaction is going to produce a water molecule. In other words, it is a condensation reaction or a dehydration reaction. Same way here, these are going to interact. What are they going to form? They are going to form a water molecule. So same, that is going to be a what? A dehydration reaction. So what are we going to see? So we're going to see something like this. I've got, let me put down, you have got a nitrogen, you have got hydrogen here, you've got hydrogen here, you have got a carbon R group, you have got hydrogen, you have got a carbon, a double bond with the oxygen is there. So now I'm going to put down this. You have seen there's a bond here. So what you're only going to see is this nitrogen, okay? That is going to be the nitrogen here. You've got this hydrogen that has remained. Now I'm going to put it down here, okay? And then you have got this portion which is here, carbon. You've got the R group. You've got a hydrogen. And then you have got another carbon. And then again, you have got double bond with oxygen, and then you have got a bond, so you are going to have again a nitrogen. Okay, this hydrogen I'm also going to put it down. Then you have got a carbon, the R group there. You've got an hydrogen, you have got a carbon double bonded with oxygen, and then this OH is ready to react with something else again. So this bond that is formed when a carboxylic acid functional group gets to react with a amino functional group is called a peptide bond. In carbohydrates, we could have called this bond as what? We could have called this bond as a glycosidic bond because we're talking about glucose, but in amino acids it's going to be called a polypeptide bond. So a polypeptide bond contains a carbon, oxygen, nitrogen, and hydrogen. Whenever you see this, just know that is, those are the atoms that are found in a polypeptide bond, and same here you have this. So when you see one polypeptide bond, just know you've got two amino acids. Now here we've got two polypeptide bonds, just know we've got three amino acids. So these are polypeptide bonds. We can also write this as CONH or NHCO. These are the atoms which are found in a polypeptide bond. That is also very, very important for us to understand. Now, another important thing that we need to get to understand when we are talking about proteins and amino acids is, since we have now understood an amino acid, how do you get to classify amino acids? So their classification. Now amino acids are going to be classified following different properties. You are going to follow if they're in a straight line or not. So some of them are in a straight line, those are going to be called aliphatic. some of them are polar. So we are going to have some amino acids which are polar aliphatic. We are going to have some other amino acids which are non-polar, but they are aliphatic. We also have some other amino acids. They are not polar, but they are aromatic. Okay. Now, these also which are aromatic can either be polar or non-polar. So let's also put that. We're going to have polar aromatic. We are also going to have non-polar aromatic. Apart from that, we also have amino acids which are sulfur containing. Well, we also have others which are imino in nature. Actually, imino is only one amino acid. Basically, this is not an amino acid but an amino acid. Now we're going to put it also together as an amino acid as part of the 20 amino acids. We also have some which are acidic and others which are basic in nature. Now these two which are acidic and basic, we are going to call them charged amino acids. Okay, remember we said when an amino acid is in an acidic condition, it's going to have a positive charge. I mean, when it is in an acidic condition, it's going to have a positive charge. In a basic condition, it's going to have a what? A negative charge. All right, so those are charged amino acids. Now these amino acids which are aliphatic means they have got a linear shape, meaning that they are straight, just like we drew the other one. Okay, we have got hydrogen, hydrogen, we've got a nitrogen here, carbon. Let's say the other group is CH3. This means this is valine. And then you have got carbon double bonded with oxygen, like that. So this is an aliphatic amino acid. But if you have got amino acid which is more like circular, you have got something like this as a group, and then you have got now carbon there, you have got carbon double bonded with oxygen, OH, you've got R group here, you've got nitrogen, hydrogen, hydrogen. This is going to be called aromatic. Why? Because it has got a ring. So aromatic is going to come on because of the ring. Now those amino acids which are polar should have at least a carbon bonded to an oxygen. This oxygen is going to have lone pairs of electrons. Okay, because of these lone pairs of electrons, it can form hydrogen bonds with hydrogens, and that is why we're going to call it polar. We are also going to call it polar. Why? Because the electrons are going to be more closer to the oxygen because it's more electronegative, so it is going to have a partial negative charge. The carbon is going to have a partial positive charge. So for the polar amino acids, they should have at least a carbon bonded to an oxygen. Non-polar, they are going to just be straight like that without a carbon. So in this case, you are going to look at the R group. If the R group doesn't have any oxygen, this obviously is going to be a non-polar. Whenever you see a bond between carbon and hydrogen, this bond is non-polar. So alanine is a non-polar amino acid. Okay, you have also glycine. Glycine has got an hydrogen, right? So if something has got an hydrogen just there, is also going to be non-polar. All right, those which are polar, we've already spoken about them. Those which have got sulfur, they are going to contain a what? They are going to contain a sulfur in them, sulfur containing. And then acidic amino acids. When you're talking about acidic amino acids, we're going to look at this R group. Remember, the characteristic of an acid is actually so supposed to be COOH. The characteristic of an amino acid is a of an acid is a COOH. So if this R group which is here is made up of this atom COO, just know that amino acid is going to be acidic in nature. The characteristic in this case of a basic amino acid is the NH. Okay, when you see an amine, it is either NH or NH2NH. Whenever you see nitrogen and hydrogen, as the R group, just know that that amino acid is going to be basic in nature. Now, imino amino acid is going to have a carboxylic acid functional group and also the amino functional group. Now, really not carboxylic acid, we are going to see the kind of group that is going to have. So these are the classifications. All right. So this is a table that can help us. Of course, the first four have not shown them here, but the first four amino acids, we have got glycine, alanine, valine, leucine, and then we have got isoleucine, serine, threonine, asparagine, and these others. So some of them we can see that these here are polar aliphatic. We have got these which are aromatic, but for the aromatic we've not stated whether they're going to be polar or non-polar. The ones on top here are going to be non-polar, though we are not able to see the first four there. Okay, we also have those sulfur containing. Now the sulfur containing, we need to know them. They are just the two of them. We need to know them. These two, we need to know them. Cysteine and methionine. We need to know these two names. Cysteine and methionine. The only two amino acids which contains a sulfur in them, cysteine and methionine. The only amino acid is only one. It is proline. And it's not difficult to know. The only amino acid is proline. We also have amino acids which are acidic in nature. The acidic ones, you can even see by the name, we have got aspartic acid or aspartate. And then we have got glutamic acid or glutamate. There are only two. It's not difficult for us to master them. We also have those which are basic in nature. We have got lysine, arginine, histidine, the three of them. Now it will be very much easier for us to see their structures to understand. So if you look at these are only R groups, these you are able to see here are on the R groups. Remember the general form of an amino acid, got an hydrogen, these are bonded to a nitrogen. You have got a carbon here, you've got hydrogen, carbon double bond with oxygen, and then OH. Now, whatsoever is bonded here, for glycine, you can see hydrogen. That means we're going to have hydrogen here. This is glycine. For alanine, you are going to have CH3, a methyl group. For valine, you are going to have isopropyl. This group here is called isopropyl. All right, that is the group that is going to be. For leucine, we have got this is going to be butylpropyl as a group, and then you have got isoleucine like that. You're able to see tet butyl there as a group. Okay, so just by looking at this, you should be able to classify which ones are going to be basic and which ones are going to be polar and non-polar aliphatic. So these which are able to see here up to this point, we are going to classify them as aliphatic. Why aliphatic? You can basically see that they don't have any chain, and then these two are going to be classified as aromatic. Why? Because we're able to see that they've got they've got a circle, not a chain. They've got a circle. That is why we are classifying them as aromatic. Now, just in the aliphatic, some of them are going to be polar, others are going to be non-polar. So, from what you are able to see, this is the R group. It doesn't have carbon, this one, this one, this one, this one. It doesn't have oxygen. So these first five, glycine, alanine, valine, leucine, isoleucine, these are going to be non-polar amino acids. Okay, and then you have got serine, you have got also the next one here after serine, which is the threonine, which also contains an oxygen. Okay, we have got asparagine, we have got glutamine. These are going to be what? These are going to be polar. All right, these are going to be polar. Now, when you're talking about polar, we are going to take these are these two, we are going to call them polar, but these other two, even if they contain a carbon and oxygen, notice that this carbon and oxygen is just not a carbon and oxygen bonded to an hydrogen. No. Okay, that is why instead of us putting them in that characteristic of just being polar, we are going to refer to them as either acidic or basic. Like in this case here which ones am I even referring to? Okay, asparagine. We also have glutamine. These two are acids. Now you can see, why are we calling them acids? Because in their group we have this thing here, the carbon with an oxygen, that is going to give them their acidic properties. So aspartate and glutamate, these are going to be acidic because of the carbon bonded with the oxygen. Remember, when you have got this group carbon bonded with the oxygen like that, and hydrogen at the end, these are going to be what? These are going to be a characteristic of acids. That's why we're picking these two as acidic amino acids. All right. Now this one and this one, these are going to be called, of course, aromatic. Right. Now this one, you can see there is an OH. So obviously we can say it is going to be polar aromatic because it has an oxygen bonded to a hydrogen, and that bond is going to be polar. But this one only contains a CH. So this is phenylalanine is going to be non-polar, while tyrosine is going to be polar aromatic. If you consider this one here, you've got tryptophan. This is also aromatic because you can see it contains rings there. You have got cysteine and methionine, the only two which contains a sulfur in them. That's why we classify them as sulfur containing. Well, we have got prurine. Now if you see prurine, this is not even the ara group. For the rest of the other amino acids you're able to see here, these things which have been put here are just ara groups. But for prurine, this is the whole molecule, and thus we are classifying this as not amino acid but amino acid. It's the only amino acid, prurine. All right. You have got also aspartic acid. Well, actually I even mixed those. So here the ones we are calling as acidic and basic, not the so these the first ones I mentioned here as being acidic. Actually, I was even debating to myself if they are going to be the ones. So these ones, they don't contain the CO or we talked about. Okay, they don't contain that CO. These are just going to be referred to as polar because of a carbon and oxygen bond which is polar, not acidic in nature. They are not going to be acidic. They are only polar. Asparagine and glutamine. Okay, the ones which are acidic in nature are here: aspartic acid, okay, or aspartate. So that one was asparagine. This is aspartate. And then the other one we saw there was glutamine. This is the glutamate or glutamic acid. You can see now the group COOH. Even this one COOH. So these are the ones which are acidic in nature. So those are just polar, they are not acidic. And we can also disqualify them from being acidic because if we only take the CO as being acidic, we also have the NH. That means they are going to be basic. Well, still basic and acidic, how are we going to classify them? They are going to cancel their charges, right? So we don't consider only this CO as being acidic. So these are going to be what? Only polar. So from here up to here, these are only polar aliphatic. Asparagine, glutamine. But glutamate and aspartate, or glutamic acid and aspartic acid, those are acidic. And then you have got those which are basic. These three are able to see here: lysine, arginine, histidine. Why are we calling them basic? Well, we have the CH bonds, but apart from that, we've got a nitrogen bonded to a hydrogen. That makes them basic. An amine group, NH. These groups, they are going to make them basic. The nitrogen bonded to hydrogen, that is going to make them basic. So this is how we are able to classify amino acids, either being basic, acidic. So these are also called polar. Basic and acidic, you are going to call them polar. And the reason explaining that is because of what we said earlier. Basic amino acids are going to have a charge. Acidic amino acids are also going to have a charge. So this is the classification, and this is the first part on amino acids and proteins.